Okay, do we have Huidong Chen here? Perfect. So next up is Huidong Chen from MGH, who's going to be presenting STREAM, which is single-cell trajectory reconstruction, exploration, and mapping of omics data.
Hello everyone. I'm Huidong Chen. I'm starting my postdoc in Panel Lab in Massachusetts General Hospital and Harvard Medical School. Today is a really great pleasure to have the opportunity to share with you our project STREAM. The full name is Single Cell Trajectories Reconstruction, Exploration, and Mapping of Omics Data. I will walk you through the following 5 aspects, like how we started this project and how our method works and the application of STREAM on both single-cell transcriptomic data set and epigenomic data set. And in the end, I will show you our software and interactive website. The rapid development of single-cell technology has brought a lot of opportunities, and many excellent work has been done, like in terms of studying the cellular heterogeneity using different clustering methods. People use like different dimension reduction method to visualize the single cells, like diffusion map, or the most popular one, like the t-SNE. But we know that the cellular state modeling is dynamic and continuous. So it would make more sense if we think about the problem in a continuous way instead of a discrete way. So that's how the trajectory inference analysis emerged. There are like 2 important concepts here, like trajectory and pseudotime. The trajectory basically describes the individual cell's progress through differentiation, and the pseudotime measures how far the cells have gone from the beginning point. It has been a heated topic since 2014. More than 50 methods have been proposed. So what else can we do here? Currently, the methods that are able to recover complex trajectories and intuitively visualize them are still lacking. And also we would appreciate some methods with mapping procedure in which we don't, in that case, we don't have to pull all of the data together and recompute everything, because that would be very computationally expensive. And also, almost all of the methods are designed for the transcriptomic data set, like single-cell RNA-seq, but very few methods actually can handle the epigenomic data set. And then last but not least, easily explorable and interactive single-cell trajectory-based database would be super helpful. So here we propose our method STREAM. I will use this example data set to illustrate our pipeline. The figure on the left shows the classical hierarchy of blood development. In the original study, there are like different ways to visualize cells. The cell can be colored using can be colored by cell types, or cells can be or some like a specific subgroup of cell can be highlighted, and they can be colored using the gene expression. This is good, but compared with the figure on the left, it's not very easy to understand, and it's not very intuitive to see the differentiation, like how HSC differentiate into different lineage committed cells. So here, like in our STREAM pipeline, starting with the single cell expression matrix, we first do the feature selection to select the most variable genes at the features to do the following dimensionality reduction. And in the reduced space, we use the newly developed method Epigraph to do the simultaneous structure learning and fitting. This is also a very nice collaboration with the NoEF lab, and the main developer of Epigraph, Luca Albergante, is also in this conference room. And if you want to know more about details, you can go find him. And finally we can get to finalize the structure. But we know that the structure is still in 3D dimension or even in higher dimension. How can we easily explore and visualize it? So first of all, we propose the subway map plot at the single cell level. When thinking of subway map, we are more concerned about how to get to different stops. We don't really care about the distance between the different sub-branches. So similarly, here we keep the length of the trajectory from the original space, and also we keep the cells distance from the trajectories. In this, then the subway map can clearly show like how HSC cells would differentiate into different cell types. The subway map is clear and intuitive, but it's not good enough because when it comes to a large data set in which many cells may overlap with each other. So in this case, it's very hard to tell how many cells we have here. So here we introduce a novel visualization, a novel trajectory visualization plot. We call it stream plot. Here we use here's the width of the branch is proportional to the number of cells. So it can we use the density to show how many cells are actually there. And once we put back the cell types labels, and we can easily keep track how different subpopulation progress, and we can easily see the composition of cellular subpopulation. And then it will be trivial to annotate the different branches. And also subway map and the stream plot cannot be only used to visualize cells, and it can be they can also be used to visualize the genes of interest. Like here I'm showing the marker gene for the erythroid branch. It's good, but giving like tens of thousands genes, probably we don't want to look at all of the genes one by one. So stream can also automatically discover some marker gene like divergent genes and transition gene. The divergent genes are defined as the ones that can best distinguish two or more branches. And for the transition gene, if we focus on one branch, and it will give you the genes that correlate well with one particular path. I will show you two examples here. Like if we look at the bifurcation around S3, stream will give you a list of divergent genes between two branches. And here I'm only showing like the top two, the CAR2 and PR3. Based on the previous literature, we know that those two marker genes are very important in distinguishing the erythroid lineage and the myeloid lineage. And similarly, like if we focus on one branch like S3 and S3, S5, stream can also return a list of transition genes. Here I'm showing like top two. We know that something beta-4 is repressed during the erythroid differentiation, and the BLVRB is activated in the erythroid differentiation. Then I will introduce another application in the transcriptomic data set. Here we introduce another like novel and unique feature. We call it mapping. It would be very beneficial to studying the genetic perturbation. Like here, we can build the trajectory using the wild type data set, and we can map the genetically perturbed cells to the inferred tree structure. And also, we can build the tree structure using unlabeled data set, and further we can map the annotated data set to the inferred tree structure to further validate different branches. And also, like when dealing with imbalanced data set, we can build the tree using the downsampled balanced data set and map the rest of the cell to the tree structure in order to keep some like rare cell types. Because of time limit, I'm only going to show you the first one. Like in this case, we can build the blood developmental trajectories using the wild type data set, and the stream can also automatically recover like two important marker genes, the GFI1 and RIF8. But we are curious, like, what would happen if we knock out GFI1 or RIF8 or both? So, like using the mapping feature of stream, we don't have to pull all of the data together and recompute everything. Instead, we can hold the tree structure and just map the genetically perturbed cell to the tree structure. So the stream plot can very easily summarize and visualize the consequences of GFI1 loss and IRF8 loss, like as the stream showed. Sorry, as the stream plot shows, if we knock out GFI1, the cells tend to go to the monocytic branch, and if you knock out RF8, it tends to go to the opposite branch. If we knock out both, it seems that cells are more trapped in the intermediate state, and it has equal chance to go either branch. And this would be very helpful in studying the cancer cells, because we can build the trajectory using the normal cells and map the cancer cell to the normal developmental trajectories, and so it would help us to understand better how cancer cells arise. And then further, we extend our stream to the epigenomic data set. We have been collaborating with Jason Buan Rostro. They provided us with this awesome single-cell taxic data set. We know that the single-cell taxic data set, it has like the number of chromatin peaks far exceeds the number of genes, and the data is way more sparse, so which makes it more challenging. Here we use the stream. First of all, we filter, in this case, first of all, we filter out some low-quality cells and using ChromeVar to score different chimeras. And then based on the this score matrix, PCA is applied, and we select like top principal components as the feature to perform the standard stream analysis. And then we can get the, like independent of the single cell RNA-seq stream actually can recover the similar and consistent blood developmental trajectories, which is quite exciting. And then stream can also automatically discover some marker chimeras, and further we can find the corresponding transcription factors, like here the GATA1 and CEBPA, which are important for the erythroid lineage and myeloid lineage. And then also like we have an interactive website available, and this website can be used as a computational tool, and it can also be used as a trajectory visualization database. As you can see, a user can easily play with the cells in 3D map. They can rotate it, and they can zoom in or zoom out. Move the pointer over the points. They can easily check all of the information of the cells, and they can also select different starting node to explore the different potential tree structure. And besides, all of the different genes can also be easily visualized in the website. And right now, like there have been 7 public data sets included in the website. And if you are interested, and if you have your analysis results, we are very happy to host your results. And also we did a massive comparison with other methods. And if you are interested, you can check out our preprint. In the end, I would also like to mention that we are also organizing a single cell trajectory comparison computation. Here the goal is to systematically evaluate different trajectory construction methods. And here Jim Fan and I are leading this computation. We are, if you are interested, if you want your method to be included in as a baseline method, or if you want to develop new method to beat all of the available method, so please check out the shared Google Doc and sign your name and add any comments or ideas in it. And right now, more than 20 labs have joined, and we are also opening all of the kind of question to the single cell community, and we hope to decide together with the community what kind of metrics should be used to evaluate different methods, and what kind of data set would be the best would be the gold standard to to validate different results. And yeah, in the end, I would like to thank my supervisor, Luca Pinello, and also all of our lab members for their great discussion and suggestion. And I would also like to thank all of the collaborators, especially like Luca Alberganti for their great contribution to this project. And also, I would like to also thank all of the funding support. Without all of their help, it's impossible to finish this project. And in the end, I would be happy to take any question. Thank you. Thank you for your attention.
Okay, so I have a question.
Oh.
So when you do your out-of-sample mapping, if you map the genetically perturbed cells onto the...
Yeah.
Do you have any notion of how good a fit it is? Like, if I have a completely different cell, will I force it on there, or do you have a metric for saying this doesn't really fit anywhere?
Actually, yeah, that's a very good question. Right now, like we have to assume the data set to be mapped should come from the same experiment. Like if there's some like batch effect there, like user have to like do all of the kind of batch correction other pre-processing step first before mapping before mapping them to the tree structure, so that in order to get some like meaningful results. Otherwise, if you just pick some random data set and mapping there, I don't think the results would make sense. Yeah.
Great. Okay.
Oh, thank you.
All right. Thanks again.